For the next 20 to 25 minutes, I'll just be discussing using CMC decision support to enhance control strategies. And really what we saw within our framework is really three big use cases and challenges. First challenge seen within drug development, building a data-driven control strategy for early and late stage development. Now from there, what we've notified and understood for risk assessment challenges was component identity, lifecycle, and scalability considerations. And this really leads to the slides that Andy showed with nitrosamines and their challenges, and I'll go into more depth in that aspect as well too. And then from there, I'll summarize the talk. Before going further, ACDLabs is about 30 years old. Um, some of you may be aware of us as a chemical drawing package in the very beginning, all the way from our NMR predictions to our physical chemical property predictions as well too. I would say in the last 15 to 20 years, we really focus in cheminformatics solutions. So the ability to open from any vendor format and ability to view that data in one interface is one of the things that's really led us to this point within Luminata as well. Headquarters in Toronto, like I mentioned, but we have offices all over the world as well too. In this case, I'm just going to go right into the use case one of data-driven control strategy and what I mean by that. So for the last little while, I've been just doing research, going on site before COVID, having discussions, and really these slides are a discussion about non-digital interpretation of analytical data, and just giving you the breadth of what occurs in the lab, still historically and globally right now. So in this case right now, you can see you can have an IR spectra, and within the spectra, you may circle certain groups, identify them, and place that information into a documentation such as Excel. Now, again, when discussing this with any of the analytical chemists that I've talked to within pharmaceutical development, you're not just going to use one technique. And what we found is that if they're looking at their spectra or looking at any other techniques such as going into Empower and looking at Chromeleon, same thing. They would peak pick using the native software within that CDS, get the peak names and get that information out. Now from there, same thing start building up within a project. For early or late stage, you start updating documentation, and most likely this documentation is living within an Excel doc. Now down below, just below the chromatogram, you may have in silico results, classes, control strategies. If you're looking at process optimization and critical quality attributes, again, living in the same location. And that's where the disconnect starts to happen. And really, when having these discussions with these different chemists globally, I started to find out, even if they're using a drawing package such as ChemDraw, BioviaDraw, ChemSketch, they started putting all of their information within this living document of Excel. And the questions that always came up from us is, well, how do you search this? How do you know what was done? Who did this? Where was it analyzed? Who has, the capability of importing or transcribing this or copying and pasting from any of those solutions? And then from there, interpretation. How was this interpreted? How was this seen? And again, from our logistics, doing a project currently, it's reasonable. But if you're looking at the life cycle of a drug and it's 5, 10, 15 years, how do you go back and find out information that's missing, specifically if it was all stored in a location that was not built to do this? So really, that's where we started looking at the aspects of creating a software platform that really distinguishes itself by looking at the challenges of data visualization, being able to take from any of the chemical drawing packages, and on top of just taking in the synthetic route, the ability to store analytical data per stage or per step. And by storing this information, to summarize and understand where this is viewed, where you can get that information, and really, notably, get that information fast and in the aspect where you know that there's no transcription errors. Now from there, going to data preparation, what would you like to do with the data? One of the key words that we've all seen in the last little bit is machine learning. But if you're living with documentation that was handwritten or placed into cells, how much of that machine learning can you trust? So that ability to just preparing that data, having it in one location, and then from there, either you doing some machine learning or even if you wanted to put into BI software like Spotfire to trust those values that when you need to just get a decision, understand that you can go back and forth with that knowledge. And really, this is a slide that Andy had as well. And these are the pillars of how Luminata really acts as a development decision support tool. Um, Cases where if we're looking at root process optimization, here discussing impurity control strategies, if we were discussing force degradation stability studies, how would we obtain that knowledge? And just know when you're doing this in the lab, most likely everyone in the group may be using a different piece when getting this information and then summarizing at the very end. Now from there, just like Andy mentioned, looking at the batch genealogy family tree. So how do we do this? I think that's a crucial thing because software platforms that are doing pieces of this aren't meant to do this, or the ability to have this type of control strategy for data is not what the strong suits of things that were existing back then. So in our platform, we really had an architecture of looking at three different databases. Really the crucial one for everyone in this call is the processes database. This allows you to look at any synthetic route, put in your impurities per step. But at the same time, this processes database allows you to store all of your processes as process maps, force degradant maps, extractables and leachable maps, batch genealogy maps. The flexibility there to allow all users to see and visualize was what we thought was very crucial comparing to how everything is viewed as numerical values. Now from there, with this architecture down of having this place where you can view all this information, you can then be able to link this with any type of compounds. So again, if you're looking at particularly a compound that was in a reaction, or even the cases that when I go down the line, when I start talking about NDMA, that ability where you can summarize and say, here's all my metadata, here's all my analytical data as well too. So I can have all my reference standards being viewed here from all the instrument vendors that I'm using. Again, we take light of this, but my background is in process chemistry. So for myself, to get that information live is always difficult. So having a platform that allows me to view this and get that understanding is quite comprehensive. Finally, as well, if I'm looking at family trees, the ability to get all my batch information, such as if I was using an EBR system, having something from SAP data lakes with all that data where I can view and understand what's occurring with my data. And that's really the crucial portions that we're going to discuss for the next little bit. Now, with that said, what does Luminata do? It really contextualizes experimental data. So like I mentioned with that one pillar slide with stability studies, within that stability studies, you can also view any of your experiments that you're running for force deg. So again, people in this call, if you're doing anything contextualizing stability, or if you're contextualizing process optimization steps, how's that being done right now? Right? How are you managing this? In the software, this allows you to, let's say in this case, in this slide, I can view all of my force degradants, what runs I want to do with it. And then once I've collected that information, automatically creates those records. So in this case, in the lower right-hand side, you'll see I'll have, let's say, an API of interest, but I have all these stages of differing time point conditions where I can view down the line what's been occurring, contextualizing that information, viewing this information, and again, not manually transcribing this information becomes very crucial for any piece of software. Now, on top of this portion, not just being able to contextualize it, but really viewing it through a bird's eye view. So you'll note in the left-hand side here, this is a project map. It's really a traffic light system of what you've done in the lab. And again, the focus here is more about force degradations or even formulations, but this could be down the line for any of the tools that you're doing within drug development. So in this case, we have our project name, differing solution states and solid states that you're running within the lab. And also if I was doing any type of drug product formulations and what studies that I've been doing are unit operations. And the beauty of this is by having a traffic light system, the user can be notified, has this been started, or we're halfway, or we've completed our study. Having that all contextualized in this one location. Now, with that contextualized ability, why is that important? Like I just said, when I was back in the lab, I remember plenty of times as a process chemist, if I'm talking to AD, the first thing I would do is I would tell them what I would have. So I have my ELN, I have a PDF, but the crucial thing and the issues that always came up was if we did see an impurity, how do we get that information out? How do I get that information, understand that further? So in this case, you see that I've really zoomed in a PDF. But for myself, when I was back in the lab, this really meant nothing. So by zooming in, I can't get that information. You can't search this. You can't repurpose this. You can't really reanalyze this. And again, if we think about the amount of times that colleagues, have those type of conversations, go on Teams, look at SharePoint, try to get the understanding, it's really time consuming because again, those portions of the project that need to be handled should have been handled right away. So you just spend all that amount of time. Like I said in the very beginning, the focus really with Luminata and within ACDLabs for a short little bit is being a platform that's agnostic. So again, Luminata is based on the Spectrus platform. And what that really indicates is the ability to import from every vendor format that's relevant for drug development for looking at analytical data. So on this slide, it just represents that I can look at micromatograms, LCMS, and any other type of instruments that are of interest to us. And again, why is this important? This is important just so we can get that view of all data from all instruments in one schematic, one view. So in this case, this is just to visualize the data for, let's say, going through early stage development. I'm looking at my process, and what the software allows me to do is have a project navigation panel. And within this navigation panel, I can have any type of layout I want to see. I can see my project, my processes, and if I'm in analytical, I can view my data. If I'm in process chemistry, maybe I just want to view all the numerical results and get that understanding of that process map. And that's what is in the central location, this ability to make this process map, this synthetic route, understanding that within the first row, if I'm looking at my reaction, that down below I can see all of my impurities, genotoxic, mutagenic, specified impurities on one view and making it live. Again, the live aspect is crucial because I can click on any of the compounds, see if I have any reference data that I've already imported within the software, and also all of the reaction stage data within this particular stage of interest for intermediate or API product. Now, again, last things for this slide, the ability to also take in those tabular views. We believe here that when you're viewing this information, people may be standoffish, meaning that they still want to view it in the specific format that they had before. So keeping that format there, where again, on top of it, everything's live, but if they wanted to look a specific way, they can as well. Now, in this case, again, I zoom in the process map, something that Andy talked about a little bit further. What's crucial here? You'll see the top row, again, is a very easy synthetic route for aspirin, but down below at every stage allows me to see the color coding of all my impurities. This, again, is quite crucial if we're looking at anything for nitrosamines. Being able to view this in any type of process. And again, with these different color coding arrows as well, it gives us an intention and understanding if it was an intended conversion, unintended conversion, what was happening in the lab. Getting the impurity fates and seeing what's occurring. This is crucial. And again, we take for granted the tools what we have, but updating this type of lab technology does help significantly, especially if you're looking at these new use cases within workflows in drug development. Now, again, I've changed my slide here, or my screen here, and you'll see that I'm showing no data. Like I mentioned, if I was a process chemist, maybe I'm not interested in that. But in this view, what I just wanted to note is you can still see your process map, but just like Andy mentioned for the work that's been done with Mirabilis and looking at those type of carryover calculations, we also offer the capability of seeing real life dynamic carryover calculations with the analytical data. Like Andy mentioned, could be quite crucial to really integrate this capability where you can see both the theoretical and observed carryover calculations in the same workspace. So from our side, having that ability would be amazing to see. And again, understanding what's occurring in the purge and fate throughout the life cycle of the processes and your batches. Now, the last thing that I want to mention on this slide is, again, we never want to change the world for anyone that's been using any type of pieces of their technology that they have. So just like I mentioned, if somebody was using Excel and they were filtering some column down, and in this case, they're filtering the column just to see all of the impurities for a particular process. We have that capability too. But the big thing that we offer is that ability to visualize with your process map, your analytical data, what occurred. So right now, 6-step reaction, you can see the impurities down below. But by filtering the table, we also dynamically filter the process map together. So just by clicking here, you'll see that now the reaction is gone, but I can see the impurity fates, and I can see every impurity that I have per stage. Now again, it's a very simple reaction, 6 steps, but for everyone in the call, just imagine having 10, 15, 20 steps, 7 to 8 impurities per stage. It starts looking like a crazy metro and looking like the tube, very tough to manage. And here it's just that ability to visualize. And sometimes that's easier to see than just going through just numerical results of any column or cells that you've noted before. So lastly, for this use case, another thing that I want to discuss is the visualization aspect, not just looking at the process map, but looking at your data. So in this case, I'm using the same type of filtering, and I'm going through the API product and maybe a particular process step that I've changed, a workup step, where instead of I've done 3 different outliers of an acid wash, base wash, and no wash. Again, you can see all those events within the current technology you may have, which is using an Excel document, but the ability to stack your plot, seeing that live, and being able to see if it was the same methods, understanding that if I do look at that acid wash, I don't see any of those impurities, that's quite crucial. Just getting those answers and understanding that becomes quite useful for everyone that's trying to obtain and understand what's occurring within their impurity control for their data-driven aspects within drug development. In this case, what I wanted to focus on, again, this all leads to the nitrosamines aspect. In the last use cases, component identity lifecycle and scalability consideration. So one of the many challenges in characterizing substances is to obtain a mass balance type qualifications for the materials. It helps to ensure process reproducibility, of course, contaminant controls, as well if your products within certain regulated industries contain impurities. There are regulatory limits imposed to those substances. So what that means, the effort to seek what we call a composite identity, an identification of all components observed in the substance that are produced in manufacturing processes, allows one to perform chemical-based risk assessment, also structure to material property assessment, then finally structure-based process controls. Basically, if you understand the source of the particular component based on its identity, you can determine where the process is produced and establish a control around it. Now, what we think is important is also the decision support needs that need to be satisfied. That is efficient identity determination processes. The process you identify a component must be efficient, also gives you effective risk management, understanding the source of how you're controlling them. Then based on the identity, what is the potential risk that material may impart? It allows you to have effective risk management. Finally, in building these, you can perform chemical-based industrial forensics. And again, from our aspect, this is something that we saw when we start going through the labs. In the next couple of slides, we'll go into details about just understanding this comprehensive risk assessment. So in this case, again, this slide looks more like, I guess, uh, a slide where we're going to go through and identify a complete unknown to a known structure. So finding that impurity and understanding what's occurring. So from the very left-hand side, if I would say this horseshoe of a slide, you have a chromatogram, and once you get to the flat chromatogram aspect and you isolate the peak in, I would say, orange, you have this new unknown impurity. How do you start to elucidate this? You find a weight, you have a formula, you collect an LCMS. Now from the LCMS, if it's process relevant, you may know certain functional groups or partial structures, but to obtain more information, you're going to have to run, let's say, MSN data to fully then elucidate it with 1D and 2D NMR to get your structures. Now, this doesn't sound bad, right? You're doing this one time, but how many times do you do that in a project? And that's really indicating in this slide. This could be numerous times. And then once you start going through this, how many times do you do overall? This becomes really abundantly clear. Where is this information being stored? Because taking the life cycle of that impurity from just starting at a weight all the way to the final known impurity structure, that's the biggest thing that we saw as a disconnect when we started discussing this with pharmaceutical teams globally. So really, this next slide is just breaking down further what I discussed earlier, just looking at aspirin and really establishing quality composition specifications, identifying the source of each mass balance term is essential. So what we're going to do is we're going to give you a use case of how the software can help in this case if you do have a complete unknown. How do you start? How is this being stored? And for yourselves, it tells a nice story because it keeps all that information in together in one location instead of just, I can imagine if you're doing this in the lab, having that Excel document where you can just delete a column once you know the structure or merge it into something else if you have a new method. So in this case, we're going to look at a chromatogram. Within this chromatogram, you have like a blue rectangular box that shows you everything's been peak picked. We have all the information. But we're going through phenol to methylphenol. We want to see what those impurities are. The first thing that comes up that we've notified is that those cases is that you may know one of the crude products, but everything else may be unknown at the very beginning. So you could either name it unknown, or in these cases, we name it with the project AZIL and then relative retention time. So once you've done that, the software keeps this within Luminata. So we know that we have the crude product, we have all these unknowns. Starting with the crude product, once you purify it, you can store all of the data, the 1D, 2D data, and also the MS all together. So now we have purified it. We have all of our standard material of that material within our compositional database within Luminata. But from there, what do we do with the rows 1 to 4 and row 6? What do we do with this information? In this case, starting off again, you have an idea, you start building a partial structure, and you'll see that in the right-hand side. Within the right-hand side, I may know a particular fragment, but for the aromatic ring, I'm not sure, and I have the Marcouche where I don't know where our functional group is going to be at. So what the software allows me to do is, again, keep storing it. I've taken it from the name, taking it from the unknown to a partial structure. Now we get to the stage where we can then elucidate this compound. Once this has been elucidated, that name that we had as AZIL, relative retention time, is now been changed. So in this case, it was RRT 1.3. Once we know what that is, we can give it, let's say, a compound name, a registration number. Again, building up from what we have from in the lab inside of the software, telling the same story. But the crucial thing when we're discussing this part of using any of the software applications is it keeps an audit trail too. So we know what's been done, what's been occurring, and what's been seen. Little things like this do make a big difference once we start looking at really genotoxic or mutagenic impurities. So again, this aspect really identifies everything from use case 1 and 2. Just like Andy was talking about, the same pillars that he's seen and just other companies with laws and waters talking about it, we notify the same things. Process-centric issues. So imagine that capability where you can see all the processes that have been done and maintain that and understand what occurred instead of just looking through, again, numerical values within a spreadsheet. And then from there, going through composition, having that ability to see and identify the drug products and understand the reaction with the excipients, having that stored in one location with your data. Now from there, another crucial thing that could come up with all these things, just like Andy mentioned, is the amount of data you're going to have to collect. It's not going to be from one instrument. All these specifications, all these values, seeing this live, stacking these plots, having that understanding. And that's something, again, that you saw earlier with my use cases. This becomes very invaluable within Luminata. Now, again, why is there this dilemma? This slide just indicates that even here, that you can have the same project and different teams, maybe upstream, downstream, but in the central location, they're all using a different piece. So think about that next time when you're working on something, how many times you work on all these different pieces, but then you have to summarize everything in one location. That's the part that becomes quite painful. And again, I always told people when being in the lab, it's a lot of paperwork, right? And that's the part where it's like, that can cause some issues here. Now, in this case, just to go through the process-centric capabilities, like you saw in my use case 1, having that ability to see all of your reactions. And within this reaction, having these connection points, having these arrows where I can see the impurity fates, really understand those processes. And later on, I'll go into the aspect of just clicking on one of these compositions to note the metadata and analytical data. But from here too, the things that I haven't mentioned is getting an understanding of the family tree and the batches of these materials. So again, in this case, if I had a nitrosamine where it was seen in one batch, how do I get an understanding what child or parent batches has it been connected to? So just like you've seen before, the crucial thing within the processes database is that that map can be anything. So in this case now, it's just a batch map of everything that I did internally, but everything that I can get externally as well too. So I can have all my metadata here for my batches. Let's say if I had it from SAP or EBR, I can have all my polymorph images here as well with all my impurity analytical data in this view. And like I said, if I wanted to get information, particularly of those impurities within those batches, how do you get it now? Within the software, just like I said about talking about impurity fates, we can see a batch fate. So if we go into batch number one, I know it's very simple, but it makes it easier. I can just say, show me every batch that it's connected to. So in this case, there's no more child batches, but it shows me right away all the connections from batch one all the way, let's say, the very beginning batch of 32966. Information that we take for granted. Again, if we're working on a particular project already, it's not that bad. But over time, and if you have to go back, how much time do you spend trying to find information? That's the crucial part. And especially if you're working with an external partner, other colleagues around the world, you don't have time for that. It becomes quite difficult just to manage. Now, the other thing that comes out of just using the Luminata is where have we seen this before, right? So if I'm looking at NDMA, I know that I have it within this process. But again, just like Andy touched upon earlier, within the software, not only can you see all the metadata and analytical data for the reference standard for your compound, but really you can actually get an understanding of all reactions that it's been involved in, but also here down below, every batch that it's been connected to. So for people on the call that are more familiar with 90s terminology, I would say this portion is kind of like 6 degrees of Kevin Bacon. And what I mean by that is that that one batch that contained this NDMA, in this case, in this image, it touched or was in contact with 30 other batches. And the question that I've always asked when I'm going through and talking to people globally is, how long would this take you to find out? So in cases of just looking through of all the different batches that were created before that had nitrosamine, how would you find that out in a timely manner? That you didn't just have to go through and really shut down everything that you're doing and just go through all of the documentation that you have internally. From there, again, lastly, within these compounds, I can also have toxic assessment views. So in this particular case within Luminata, we can take values that are coming in from other external software. So in this case, we have the ACDLabs platform of Percepta, but if you're using LASA and you have that information, that can be stored here too. So that ability to just be a one-stop shop and understand what's occurring is quite crucial where you just want to get those values out on top of, again, summarizing where it's been seen, all the data you have, also getting your tox assessments here as well too. Finally, if I wanted to get a read across and view, let's say in cases where I'm viewing all my impurities and seeing all my theoretical results compared to my observed results that I've taken for my ADME tox, you can do that here as well. Quite crucial. Again, little things that we take for granted because we use all these different platforms, but at the end, when we're coming up with that knowledge, it's still coming down to writing it down or copying and pasting somewhere. Well, here it's all in one location. So again, I'll recap in the next two slides. Well, what really I wanted you to get out of my portion of the presentation is how the software here can look at three different pillars. Being a process-centric, having that capability of looking at a use case where I can view, understand what's occurred within my process, my batches, my force degradation maps, stabilities, and having that connection with all my impurities, my genotoxic impurities. Compositional-centric, really, again, when we look at aspects of looking at impurities and we're looking at these specified impurities, how do you start off? How do you understand where you're coming from an unknown all the way to known structure? where's that being stored? How do we get consistency? What's the biggest thing that we always see in the lab? You know, product gain, efficiencies, doing things the same way over and over again, right? Finally, the ability to just get a read across for all your compositional information as well too. So um, like you saw earlier, again, the tool here is just the ability to just merge all information, really get that understanding from all the support and software applications that you're using and place it in one location with data. And within this data, being able to just understand what's occurring to better suit yourself when something does come up, with this case with the nitrosamines, where you can handle it and not just start scrambling and trying to find information, but being able to store that in one central location. Again, from my side, wanted to thank Claire again, Andy, for this time. I really appreciate it. From my side, Jesse for the poll questions, and also for looking at the slides, and also my boss, Andrew Anderson.
Great presentation, very, very interesting. Andrew, did you have any thoughts on anything that we discussed to maybe kick things off?
Yeah, I mean, one of the things that I'm still really, really interested to understand is, how can we create that holistic environment with all of this? I mean, I understand how the tools you've worked and from the examples you described, are still a bit that is missing the right word, but I'm intrigued as to how we might be able to take all that information you described as spectral data, the batch tree information, the tracking of impurities, and ultimately tie that into, say, something like Mirabilis, where what you would do at the moment in Mirabilis, you would take the same route, you define what those impurities are, and you basically do the calculations to work out what the fate and purge of those impurities are. Wouldn't it be great if it was possible to sort of link that all up in a way that I can look at the actual real data that I mean, in some cases with the purge calculation, you may say, well, it's option 4 control, it's a mutagen, don't need necessarily track it. But in those cases where we do want to track that further to understand what the real fate and certainly the real purge calculation is in terms of what you experimentally see, the ability to be able to sort of being able to actually be able to look at that and try to understand across different, it may well be that you do that across several batches. Is there one batch that stands out where actually the predicted purge and the measured purge were very close together, where in all the rest, as you would expect, of purge calculations that actually, the measured purge was significantly higher? Is there one batch that stands out as an outlier? And why might that be? What could you interpret? Could you go to the batch tree and say, well, actually, you want to track it back, and I track it all the way back and find out that there was for instance, a deviation or something like that. I mean, at the moment, the only way I'd find something like that out that would explain an outlier would be going through a sort of fairly tortuous conversation and just hoping somebody remembered as to why the one batch is different to another. And, that's far from ideal. I mean, it can be done, but it's, it hit and miss at times. And it certainly doesn't fit in well with some of the kind of practical problem solving exercises that are often done as well, which through not having that kind of hierarchy and tracking that you've got, is that compiling all the data to do those kind of problem solving investigations is tortuous. I could see that this could save an awful lot of time in that respect.
Andy, we've had many conversations. I think we're on the same page. This holistic view, getting that understanding in those platforms I can see that's the next conversation piece that we get, the right people involved, get us in a room. And that's what I'm really hoping for. I just came back from Italy last week, and, the ability to just, see someone and just discuss this through and find this out. And again, we talked about this, but you'll be amazed how many times I hear from other people as well, other scientists who are like, hey, Joe, can you do this? And I'm like, that's something that we need to discuss. So yeah, let's definitely offline. Let's get in that room, get in those places, and go from there.
How does the implementation of continuous manufacturing challenge us in tracking slash purging nitrosamines? What are your recommendations about nitrosamine assessment in a continuous manufacturing scenario? This is a really interesting question. Maybe we'll turn this to Joe first, and ask you about the like if that's something that you can manage in Luminata, and then Andy, if you have any recommendations on how you can think about it too.
Like I said, well, from Andy's talk, we know the amount of data that's coming out of here. this is going to be a huge thing where being able to just store this information, I know from our end, it's taking all these, let's say, longitudinal points, understanding that, viewing that, filtering it down, and being able to store and understand what's occurring. It's the biggest strength that Luminata has, right? And if we can do that with the continuous manufacturing, getting those probe and information and being able to place it there, that will help out. And the question for the user right now would be is how is that being done right now? Like, how are you viewing this information? How can you view this information? And like Annie and I just talked about the last question. It's like, is this something where one person could get their hands on, or is it this, again, a conversation that we need to go on Teams? We need to have a weekly meeting. Like, we need to figure this out. And that's the things I would love to ask. Maybe the person asks a question after in the booth, or we just go through and have a conversation about it as well.
Yeah, I think that there's probably solutions to do this in various ways, because they're not going to be using this one continuous stream of data. They have to chunk it up and use it in some fashion like that. Maybe, Andy, though, you have something to add and to think about doing nitrosamines assessment and continuous manufacturing.
I mean, it's not a question I've had asked to me up till now, and I'm sort of sat here in a little bit trying to think, well, what is the questions that I need to ask myself to be able to answer that question, if that doesn't sound silly. I'm trying to think, well, what would be different about a continuous process, and therefore what are the factors I'd need to take into consideration? And I think it is definitely one I would have to take away and reflect on. It's it's one that you think there should be an answer to. I'm trying to get it in my head in terms of, is it more of a risk or less of a risk? Well, I guess it depends on what the continuous manufacturing process is and what the steps in the unit operations are that's involved in this. Is there any way that through continuous that you get a gradual build-up, either of a nitrosamine or maybe even the precursor, in a way that you wouldn't get in the batch process? So there's lots of sort of different questions you'd need to kind of ask yourself before you could think in terms of how even to answer that, beyond the fact to say it's a really interesting question, and we probably should be able to answer it if that.
Well, I mean, one of my first worries for this is that if you are using a continuous manufacturing process and you have a risk of nitrosamines, like, I mean, obviously, like, different processes have different risk for it, particularly given how low those thresholds are. That seems like it could lead to big problems, like if you do get into trouble with it, because you're going to have like a long lot of batches that are suffering like those issues. So maybe that's just a question of developing a process that is thoughtful about the sorts of risks that you're going to be encountering. We did have some other questions. I don't know if we are still hanging out waiting for Claire to kick us out. But yeah, maybe we can add another one. So one that Joe, you mentioned early on in your presentation, talked a little bit about, was formulation was one of the things that you mentioned as a function of Luminata, and that was something that came out very clearly in Andy's presentation about excipients as one of the sources of risk in n-nitrosamine creation. Did you have any thoughts as to how a tool like Luminata could help in assessing risks for formulation in nitrosamines avoidance?
Yeah, of course. Again, I'd really try to focus on a couple of use cases, but the ability of, again, one of the things looking at excipient studies, but also the ability to store that and look at those unit operations and view that all in one location, right? I think when you're doing formulations and some could be done in LIMS, some's just done in differing instruments, it's the same disconnect happens again, where if I'm doing dissolution studies, I'm using something else. If I'm doing, again, excipient studies, I'm using another piece of the software. So how do we actually reconnect that? And in this case, have that ability to see that API or see that excipient, just like in those cases that Andy mentioned, and really relieve that or see that and visualize that before, again, it becomes more of an issue. I think that's the biggest thing, is that no one wants to be reactive at this point. Reactive is causing a lot of problems here. And by being reactive, and like Andy's talk where he's pointing it out, where it's like, we need to hit this specification. Well, what happens if you don't? If this wasn't an issue, how can we really mitigate that? And really, Luminata just allows you to store that piece as well, and having that and being able to report that. And even from further on, if our connections that we talked about with just having machine learning or using those type of abilities, that becomes quite crucial here, because We want to envision what's going to happen soon. But just like Annie mentioned, what happens if they take away the Ames test? Like, what are we doing? You're right. We need this to have this now. So that's how Luminata can help out. Again, that ability to just have that storage and visualize and be a one-stop shop where they can view this and carry on as well.
Now, Andy, are there any, for formulation specifically, are there best practices that are being developed that you're aware of, or advice that you'd give for people who are maybe concerned about this?
I mean, there's a lot of different dynamic factors that relate to this. I mean, there's some general simple things. I mean, first and foremost, this idea at the moment that you could formulate with nitrite-free microcrystalline cellulose or something like that is, well, that's not practical, so that's not going to happen. I mean, we're working with excipient manufacturers to look at ways we might be able to reduce the level of nitrites in certain excipients, but I mean, we live on a planet that, has an atmosphere of nitrogen and oxygen. We're not going to get away from that anytime soon.
We're working on it, but we're not very much progress just yet.
Well, yeah, unless we just replace all the oxygen with carbon dioxide by burning too much fossil fuel. But yeah, but I mean, so there's, we have to be sort of practical and pragmatic of where we can go. We can understand what the relative levels are. Is there something we can do in terms of, adjusting the relative proportions of some excipients? I mean, we always had this kind of general basic thing that says, well, we have this percentage of a particular filler, and if that particular filler is a nitrite, contains a relatively large proportion of nitrite, do we need to change that composition of the formulation? We could go even further and say, could we include scavengers in that formulation, like amino acids and things like that? There's things we could do in that respect, but would you do that for a product that's already marketed and file that as a variation? I think extremely unlikely when you consider all the baggage that would go with that. You'd have to repeat your stability studies. You probably have to repeat your dissolution studies, maybe your bioequivalence studies, and so that's not a trivial thing to just lob in an extra excipient if you've already got a product. New product might be slightly differently. If you're going at this for the first time with developing a new novel formulation, maybe you've got a bit more flexibility, in regards to that. You could choose your manufacturing process. If you think there's a major risk, you could go with dry granulation rather than wet granulation, which we know is a slightly lower risk. But it's quite difficult to sort of, say, I mean, I guess what I'm trying to say is these are all various options. I don't think it's possible to sort of say that, well, if you did this, this, and this, that would immediately, alleviate the problems. I think you just have to be aware of all of these, call them tools in the box that you could possibly use for this. But I don't think you could go as far as to saying systematically, well, if you did this, this, and this, you would always automatically avoid the risk, because even then you come into, well, what's the drug loading? Is the drug itself a secondary imine? Well, in which case, most processes are going to present a risk, even dry granulation in that scenario. But yeah, there's all of these things and variables you need to think about. And the other variable and dynamic we're really struggling to get control over at the moment is what's an acceptable level. Because most of these formulation-related problems with the level of nitrite we've got go away if you set acceptable limits that we would consider to be more appropriate from a safety preclinical perspective. So, all of these kind of things come into the mix.
Yeah, I mean, that's definitely a theme that came out, I think, in multiple of the presentations, of this question of, until we get a handle on where the acceptable levels, like the limits are really supposed to be, like what makes sense there, that there's a whole lot of like just trying to figure things out and what's the best, and it's hard to kind of solve those.